Okay, so today we're talking about passive and active transport. Generally, we're talking about permeability, which is how substances flow in and out of the cell. Or another way of saying that is how substances pass through the membrane. More generally, we're just talking about the movement of molecules through solutions, but these are the ways in which they do it. All right, so there are 4 types of passive transport. Can you name them all?
Osmosis.
Okay, good. We'll put osmosis there. Number 2. Good. Diffusion is number 1. This is kind of like Family Feud.
The kidney one with pressure.
The kidney one with pressure.
Facilitated.
Okay, that's a different one, but still one. Facilitated diffusion. And Brigham, it's on the tip of your tongue. A. Okay. U-L-K. You got an L in there. Bulk flow, good. Man, you guys are good. All right. So bulk flow, or you could also call this filtration, okay, which occurs through the kidney, through all of your capillaries. That's how fluid is pushed out of the capillary pores, okay? All right, so let's talk about these on here. Passive transport, by definition, what does that mean? Right, it does not require energy.
Is that any type of energy or specifically?
Well, in the cell, energy, the usable form of energy is ATP. But yeah, not all processes use ATP, but biological ones do. So, passive transport does not require ATP, does not require energy individually. Diffusion, this one's really easy to envision, understand, everything. When you have a concentration of any substance in a solution, what's going to happen to these molecules? Exactly, they're going to spread out in all directions until they are evenly spaced out. So this is going from greater to lesser concentration. Okay, or another way of saying that is it's going with the concentration gradient, okay, in a natural way. And this does not, and actually it is just the concentration gradient changing from greater to lesser concentration. Okay? So, some examples of this. Someone squirts some perfume in the room. It's going to be concentrated around that person, but eventually it will diffuse throughout the room to where everyone can smell it, right? And over time, that perfume itself will be lost through the air. It'll diffuse, and you won't be able to smell it even in concentration, okay? All right, there are grosser examples, but that's one of them. All right, okay, osmosis. In order for osmosis to occur, what do you have to have? Okay, you have to have water, good. What else? A membrane, right? That is semi-permeable. So this is my red line, this is my semi-permeable membrane here. Okay? Your phospholipid bilayer is another semi-permeable membrane. What is the semi-permeable membrane that we used in lab a couple weeks ago? Yeah, the dialysis tubing, okay? So, semi-permeable meaning it is somewhat permeable or permeable to some things but not other things. So, in this instance, it is going to be permeable to water, but not permeable to this green molecule that I'm going to put in here. All right, let's say this is glucose. All right, if this permeable membrane was not here, semi-permeable membrane, what would happen to the green molecules? Spread out, right? They would diffuse. All right, but because it can't get through there, these little pores are too small, so every time it tries, it just bounces out, okay? So instead of the molecules spreading out and creating a more even distribution of, let's say there's a few over here, more even distribution of those molecules, since they can't do that, what's going to happen instead? The water, which can go through, is going to try and even things out. Okay, and so what will happen as water goes through the semi-permeable membrane, this water level on this side will increase. This water level on this side will decrease until these molecules are at an equal concentration to the ones on the other side. Yes?
So there's also like different type of molecules on the other side, and they're not able to pass through this way.
Right, they can't go through that way either. They're going to bounce back. But there's less of them on this side, right? There's less of them on the right side. So as the water moves over to the other side, it's going to make this concentration more dilute and this concentration less dilute until they are equal. Okay? All right, now when we started, we have two solutions here, right? And we can describe them in relation to each other in terms of their solutes. Okay, the one that has more solute is called hypertonic. Right? And the one that has less, I'm going to draw a few more of these in here, is hypotonic. Good. And so the water is always going to flow from the hypotonic solution to the hypertonic solution, which is what we showed. And that'll continue until they are both. I'm going to add another one. Hypo means less. So if it has more solutes, it's hypertonic. Less solutes, hypotonic. All right, that all easy? Understood? They're both equal, right? And the other term for having equal concentration is good, isotonic. Okay? Yeah, good. So yeah, isosceles triangles have equal angles at each point. So iso means the same. Okay? These can be terms can be confusing, but just remember that tonic is referring to the solutes. Hyper means more. Any questions? Yes?
So on the exam, how would you test us and
Oh, I could ask which way the water is going to flow. I could ask which concentration is hypertonic or hypotonic. I could ask, things of that nature. Okay, our third example, facilitated diffusion. Let's draw the same system we had last time with a semi-permeable membrane and a hypertonic solution and a hypotonic solution. And in facilitated diffusion, same system, except in this one, we also have a channel protein which then allows diffusion to occur. So rather than the water having to go through, you have these channels set up which change shape in the presence of the solute to allow it to pass through the membrane. And in that way, they will go from greater to lesser concentration until the two solutions are isotonic. Now, the water won't change at this point because the solutes have a way of getting through.
Okay, it's fast enough that you'll do that.
Yeah, maybe there'll be a little bit, but when it equilibrates, they'll have the same number.
Reach equal.
Yes. Okay, yes.
So I'm just confused. So basically for the facilitated diffusion, you need this channel protein so that the molecules on the left and then go over here to the hypertonic area, correct?
Right, so they're going to go from the left, which is hypertonic, to the right, which is hypotonic, until they are isotonic.
And that only occurs when they're going from hypotonic to hypertonic when there's those transmissive channels.
Right, if it has some sort of channel to go through. Otherwise, they aren't going to fit through. And that's when osmosis would occur. All right, this last one, bulk flow. Okay, I'll give you an example. All right, this is very simplified. But when your heart beats, it's creating a pressure, okay, which is going to push on the outside of your arteries, okay, and cause them to expand. It's going to create a pressure. It's forcing this solution into this area where it doesn't fit very well. When the solutes, I'm going to draw little solutes in here, arrive at the capillaries, they are forced out. The solutes and the solution, and anything that can fit through there will be forced out. Kind of like when you turn on the hose and then the water comes out the end. You have water pressure when you turn it on, and now it can exit out of the hose. Especially, actually, this makes a lot more sense if you think about those hoses that are just like in the garden with the tiny holes in them. So that's the same sort of system. Now, it's a little bit deceiving because at this point down here, yes, you aren't using ATP, but in order to create the pressure, the heart itself, it used a lot of ATP to do that, right? It did use energy upstream, but at the site where bulk flow is occurring, there's no ATP needed. Right.
I don't know. So it'll continue to flow down too, just not pressurized.
Yeah, so what actually happens is yeah, it's a continual flow. There's less pressure on this side than on the other side. But yeah. Okay, that's passive transport. Any questions about that? Okay, active transport then means what? Yes, requires energy in the form of ATP. And what does it mean in the terms of flow of concentration of solutes? So remember, passive was from lesser to greater, from greater to lesser concentration. So what is active? Lesser to greater, right? Okay? What's going to happen if you are continually making a greater and greater concentration on one side? They have pressure, yeah. Well, those solutes will also want to diffuse back, right? So this takes a constant amount of energy. If you want to create a concentration of something, it's going to take energy to keep it there because it's naturally going to diffuse. All right, so one of the ways this happens is through carrier proteins, and there are 3 types. Well, there's lots of different types, but there are 3 types that we talked about. A uniporter. In a uniporter, you're just taking one substance and transporting it across the membrane. Okay? So an example of a uniporter is a hydrogen pump. And we'll talk about hydrogen pumps later. All they do is they take hydrogen ions and they pump them across the membrane using ATP. All right, a second one, symporter. Oops. Okay, where you have 2 substances going in the same direction. Oftentimes you'll have a hydrogen pump where it creates this concentration of hydrogen, and then that hydrogen will go through a symporter and carry something along with it. So the hydrogen will naturally diffuse through, and then it'll carry something with it, such as glucose or something. Okay. And then the last one, an antiporter, where 2 things are going in opposite direction. So an example of this is sodium potassium pumps. Okay, and these are very important for maintaining gradients in nervous tissue. Without these, your nerves don't work. And they pump sodium one way and potassium the other way against their concentration gradient, which costs ATP. All right, bulk transport is transport of substances surrounded in a membrane or using the membrane somehow. So you have 2 main types. Exocytosis, which means it's going out of the cell. And endocytosis, which means it's going inside the cell. Okay, and I'm just going to do a little drawing of this. Basically, what you have to have in order for this to work is a vesicle that has something in it. So let's say this is a lysosome and it has a bunch of little, amino acids in it. And it's within this larger cell, or larger membrane, the plasma membrane. And if exocytosis is going to occur, what's going to happen is this membrane is going to fuse with this membrane. As it does that, it exposes the insides of its cell, and then that just leaves the cell. Okay? Endocytosis then is the opposite. Let's say we have this packet full of water. And it's going to go into this cell. Well, this membrane is going to then fuse with that membrane. And then that water is going to go inside the cell. If it is water, then the process is called pinocytosis. And if it's an energy molecule, like some food of some sort, such as, let's say we've got a bunch of glucose molecules in here. Then it's called phagocytosis. pH. So if it's anything other than water, it's phagocytosis. So this is also used to describe how white blood cells will surround a bacteria, surround it in a lipid membrane, and then enzymes will destroy it through phagocytosis. But this requires ATP. So in order to get this to work, you have to have a bunch of little ATP molecules and proteins. All right, what did I draw here? Is pinocytosis endocytosis or exocytosis? Endo, yes, going in. What about the little amino acids? Exocytosis, right? So it's getting moved outside the cell. Here you have a cell that's important for making proteins for other parts of the tissues. It may constantly be phagocytosis or exocytosine. I don't know if that's a verb there, but things outside of itself. Yeah. Uh-oh. Okay, yes?
So is it constantly just going through these cytosis phases, or is it does the cell just go, okay, I need to do cytosis phase?
It depends on the function of the cell. So let's. Okay, last thing we're going to talk about is cell signaling. So throughout, especially multicellular tissues, there is communication which occurs from one cell to the other. One of the ways is through a hormone, okay? And that is a endocrine signal, right? So there are three types that we talked about in the lecture. Endocrine means it's going from one tissue to a different tissue in the body, okay? There was also paracrine signaling, which is going from one cell to the other inside the same tissue, and autocrine, which is going within the cell. Okay? This one I'm going to talk about is, or some examples of endocrine signaling. So if in an endocrine signal you have a signaling tissue and a target tissue. And the fluid which it's going through to reach the target tissue is what? How does it get from the signaling tissue to the target tissue? How do hormones work? So you produce a hormone in the brain, and then it goes to your ovaries. How does it get there? Not the nervous system. Blood, good, right? So through the blood. All right, so insulin